So, hello and welcome to the NFDI talk. My name is Kurt Wiljes and I work at the NFDI Association's office. In our NFDI talks, scientists from different disciplines present exciting topics about research data infrastructure and research data management. All talks are live streamed and saved on our YouTube channel. After the talk, we invite you to ask questions and to discuss the topics. This interactive part will not be recorded. So in today's talk, Björn Usadel will present FAIR Digital Objects, Infrastructure Agnostic Research Data Lifecycle in Agro-System Sciences. Björn Usadel is one of two directors of Bioinformatics Department at Forschungszentrum Jülich and heads the Omics and Database Bioinformatics Division. His work bridges molecular biology and computational methods to advance plant systems biology. Björn Usadel serves as a co-spokesperson of the NFDI consortia FAIRagro and DataPlant, and he heads the FAIRagro task area for infrastructure services. So, Björn, we are looking forward to your talk. The stage is yours.
Yeah, thank you, and thanks for giving me the opportunity to have this nice outlet here and to be streamed on YouTube. Yeah, I will not repeat the difficult title, but the difficult title is representative of what we are working on, namely relatively heterogeneous data. So if we work in FAIRagro, we work in a domain that is multi-tiered and has multiple key challenges. And we want to secure food nutrition, we have to battle climate change, we have to deal with biodiversity loss, we have natural resource degradation, pollution. There is, of course, a target for the 2030 strategy. We need to reduce pesticides, nutrients, and increase organic farming. So we actually span ecosystems, biomass, and insects. But if you look at this, we also go to global scales where we look at glyphosate as a pesticide application, nitrogen surplus, and that describes well the different disciplines and the different data sets we encounter in FAIRagro. So we actually have to bridge different scales, disciplines, and data categories, as I will show you. We want to have sustainable solutions for the science, and this needs an agro-system approach. For crop productions, ecosystem, landscapes, we want sustainable management, we want to have a good development, but we need an understanding of the plant, the soil, the environment, and their interactions, but also look at the cross-scale processes. What does that mean? Well, the point is we actually go from very different scales. At the very bottom, we have molecular genes and genomes. We have the plants going into crop trials, farms and fields, landscapes. We even look at regions, as you saw before. And these are very different scale, going from the microscopic and molecular data all the way up to regional data at a much higher level. So FAIRagro has a lot of subjects and disciplines, namely on the plant sciences, soil sciences, environment, and actually covers everything from agronomy to modeling, remote sensing, molecular data, plant nutrition, to name but a few, and is growing to have more disciplines covered even. As such, FAIRagro needs to deal with a lot of data categories as well. We have time series data, we have this geospatial data, we have farming data, we have also sensitive data because, of course, sometimes a farmer might be a company, or if you fly over a field, that can be quite sensitive data. But we have lots of omics data at the molecular level as well. And we have legacy data and databases, which I'm going to describe to you in some detail and how we cover this. This actually gives us quite a lot of research data management challenges. We have the legal questions at the very bottom. These are tackled. I'm not going to talk about this today. But we have data quality. How good is my data? What kind of data can we trust? And Are the data that we have even plausible? For example, if I find like 500 tons of maize on a very small field, which is not even a hectare, this is probably wrong. Is my data set even complete? And that is something we will tackle today. How can I in the end publish my data sets? And what kind of method was I using? And for that, we have lots of different standards on how to deal with the data, but also on how to describe the metadata and go for this. At the very bottom, we have, for example, data site, very well established. But specifically, we have, for example, myopy, which describes very well how we should describe such a plant phenotyping experiment, for example, and that gives us a lot of metadata. And at the end, we want to ask, where can I publish my data? And where can I actually find the data back from other ones? And as you can imagine, with the heterogeneity of data and the legacy data we are not coming from nowhere. And even before the NFDI, there were databases. And you find data in OpenAgra, IDAL, Publiso, Bonares database, which was a lot about soil data and is quite an established repository already. But the question is, as a user, where do you look? Do you look in Bonares? Do you look at the IPK-DAL database? Do you look at both? Do you need to look at other databases? And how can you bring this together? This all resulted in something we called data management could be unfair for users. It might be difficult for users because not only did you have to deal with things that people that actually made the data keep in mind, like all these different standards, because this you hope will be provided, but you need to know where to look. And this is where FAIRagro came in a couple years ago to no longer constrain required advances in agro-system sciences. FAIRagro was built around use cases to actually ground FAIRagro into the use of the things that actually are specific problems, ranging from breeding, of course. This is very important, of course, in the agri-science systems, but also working on sensor systems, pest and diseases, longer-term experiments, agronomy, and we wanted to have integrated modeling, to just name a few. And you see that also the use cases span a broad range of scales, both on the molecular and on the plant data, but also bridging to some other disciplines when we talk about sensor data, which connects geospatial data, but we also need to understand what these sensors bring. And because we have these use cases, and because we have all these different tools in the plant sciences in FAIRagro, it is not surprising that when FAIRagro started, we had a lot of databases already. These were, well, let's call it maybe legacy databases, but it's also something that FAIRagro could deal with and actually start out, so that FAIRagro actually had some data to start with and was not starting with nothing. But sometimes, then, of course, you need to find a common language. So just to name a few, we have this IDAL PGP at IPK. I already mentioned the Bonares database, but there is OpenAgra, there is the JKI data group. There's many, many different databases that hosted data, which is important for agri-science systems and which you wanted to explore, but which database to use. And to tackle this, there was actually a use case, and we're looking at this in our research area 4 in FAIRagro, which is headed by Matthias, and looking at core databases to actually develop a common ground. And we started with 5 core data sets, which have quite a lot of data, and which we could use to build a common middleware. But now if you look at that, if you just look here at the metadata standards, you see Dublin Core, DC, Dublin Core, Dublin Core, Dublin Core, Dublin Core, all of them have that. And on top of that, you have DataSite, you have Mods and Spire and other things. And of course, not only do we have different data standards, but also the API was adapted to the database that was there. Some have REST access, Some others have other accesses, but you see, once again, there is not a common ground. And if we go for metadata standards, we have common ground, but this is just mostly doubling core, so a very low set of metadata. The question is how to move ahead from this towards something which is completely integrated. And if you think about it, this is actually a mini cross-discipline use case, as we see in multiple other NFDIs as well, which are relatively heterogeneous. Also, if you look at the NFDI, we always say, yes, it's broadly and spanning and very heterogeneous, but we often have the very same problem. And not only in the whole NFDI, but we have the problem of different scales, different databases, and we started with different standards also in FAIRagro. And because we have that, by looking at that, we have an opportunity to actually develop solutions that we can use, not only within FAIRagro data plant, I will come to this, in the life sciences, but also beyond, by actually bridging and developing infrastructures that can be used to share, to reuse, and to make data interoperable. So in FAIRagro, we had something which we call minimal standard, a basic middleware concept. So the first question as a user, of course, is, where is my data? Where is the data that I'm looking for? And how can I get the data so that I can get this? For this, of course, we cannot just take all these legacy databases and make them new and put this all in a big, big data chunk. We thought about it, but of course, that is not good. And it's also what happens then in the future if we want to connect a new database. So the idea was to have this basic middleware concept where we actually have the APIs. We can harvest data from this different databases. We need discovery services, of course, and our middleware, which we actually share with GitHub, is then used. Okay, so we have all this data. We get metadata and data sets from that, but only on a high level. So we don't extract the actual data from the databases, but we just extract the metadata for now to expose it in this FAIRagro search and inventory portal, which is based on the Dataware's technology. So we harvest this, put this in here, and like this, we can actually go ahead and ask what kind of data we have. And that is actually just coming live. So you can search now for data. For example, I searched here in this search and inventory portal, which is hosted by the ZBMed partner and programmed together with us. We search for nitrate and you actually get these different data sets, and this keeps on going on. You can also filter and build some queries in here. And what you see here is a cover crop derived microbial biomass, nitrogen uptake, and you see here the link. This is Bonares. This is one of the databases. And now if you actually click on this, you get more metadata about this data set, and you can access this data set because you have a direct link to the database. However, the data still resides in the database. So in this case, it would still reside in the Bonares database. And while you can actually learn quite a lot of it, here you see the link, you get additional metadata that you get the doubling core, of course, and you get more data which is being normalized. The question is, sorry, and we can also search for repositories. So of course, we have a limited number, but all of them are tagged with different keywords. So you can look by keywords as well. And for example, yeah, here we selected the Bonares one because it just has a lot of keywords. So you can look for data sets in food safety, whatever you think is important for you, and you get these different databases. Now, these different databases and data sets allow you to get your data. But I already told you, if you just look for nitrate, you might get dozens or even 100 data sets. So what do you do with this? And did we forget something here? So we can now, at this time, and remember we assert phase repository, we can find data with metadata and we can reuse it. And we had just this data science talk a couple of weeks ago by Professor Schimler. We actually have a similar approach. You can search for things, you can find your data, and you can reuse it, because now you have an ability to find it, and we have basic metadata. But what happens if we actually want to do more? What is actually the interoperability? What about the accessibility? What about machine actionability? What to do if we want to compute on this data? We want to machine learn on this data. What if we want to get data together? And for FAIRagro, this is the extended FAIRagro middleware, where we're actually working very hard on this. And that is the, for example, the scientific workflow infrastructure, where you can actually send your data and check this. And you see that this data set, this is now very different. And you see this is embedded together with the data plant, because we're using data plant technology, and of course the whole NFDI. And here the idea is the research data infrastructures, the same ones as before, now registered to the middleware, to this extended middleware, and can communicate, of course, still with the FAIRagro Search and Diverse Inventory portal. But now we can do much more. So the idea is what we are currently working on, and we will show you some results of this, the FAIRagro scientific infrastructure, but we need something to communicate, and these are RO create, research object create, FAIR digital objects, so that we can go across, but also talk to other data sets by using NFDI data hubs, communicating with the plant data hub from data plant. And this is here from this publication of Brizuela. And how and why do we want to do that? Remember, NFDI always strives for an integration. And the question here is not that everybody wants to cook their own thing, but here the idea is because data plant actually started 2 years before FAIRagro, it developed technologies that can be reused, reused, and be used for additional data sets and for the benefit of both FAIRagro and data plant and additional ones. So within the life sciences, we are also working together not only with data plant, but also driven by this. There's a whole group of life sciences working together, working on FAIR digital objects, specification and infrastructure driven by data plant, and working on FAIR digital object analysis and workflows. So why is this important? So let me remind you, the databases we have so far have as metadata standards, in the worst case, just Dublin Core. With Dublin Core, this is good to have. It's a minimal standard. It's a good start. With this, we can search. But we need more, because we need actually to know what has happened, what has an experimentalist done, and what kind of data does the experimentalist have to actually execute, for example, in the SciWin, to put it into other infrastructures, or to communicate with other infrastructures. We need more metadata. So for this, of course, we need ontologies, but also importantly, we need systems that are standardized and can be reused. So this is where the connection with data plant came in, and this is actually a connection going even further, because also data plant did not build things from scratch just to have something new. You might know this famous XKCD comics. We have 10 standards. This is not good. Let's make a new. Now we have 11 standards. No. What we want to do is integrate standards that are usable and use them for FAIR digital objects. And this is building also on something called the Research Object Crate, which is developed by Carol Gobler and colleagues. And the idea there is that you actually have a DOI, you have standard operation procedures, workflows, you have software, you have models, presentations, article, and data, and you put this all together. And you get an integrated view. And this is called Research Object Crate. This is not necessarily fair yet. You add a little bit more on this, but the DOI already makes it fair. There was a discussion whether or not this is fair, but the important thing is, yes, we can, of course, make it fair. And importantly, it is fair in the way we are doing it. Once again, it's good that we had this talk by Professor Schimler about FDOs already. And here the idea from data plant came in. The data plant developed something called an ARC, an annotated research context, which is an FDO, and it's an ARC RO-crate profile. So it is an RO-crate, so it's completely compatible, but it extends this. So we have data, metadata, protocols, operation, persistent identifiers, and workflows. And importantly, all the necessary ontologies that we can actually use, reuse it, go across with this, and actually apply it also in the FAIRagro domain. The idea is to collect data, to preserve data. We have controlled vocabularies. And because you have this all together now, it's this idea. You can, of course, search it with Google search. You can put it into Dataverse if you want to. But now you have all these additional assets. You're not only restricted to Dublin Core anymore, you get more. And you can find adapters that fit to this. You can get a data publication, which is already done by data plant, and for the molecular data and other data sets, you can send subsets of the data to specialized endpoints. Why would we do this? For example, in the molecular field, lots of people will go to the ENA, at the EBI, the European Bioinformatics Institute. It is good measure to put certain data sets in there. Proteomics data goes into proteomics databases. Our RO-crate, this ARC, actually has all this data and metadata. It's a superset now. From the superset, we can take data sets out and put them into domain-specific repositories. For FAIRagro, however, we have this data together. For data plant, we have this data together. But we are a nice player. We play with our colleagues from England. We play with our colleagues from the United States. They get what they need and what they want. We can slice it out or transform the metadata in the way we want. And once again, data plant did not reinvent the wheel. Data plant built on something which has been around for the biological sciences for quite a while, which is the ESA standard. And that actually goes back to more than 13 years already. And the idea is always, if there's a good standard, use the standard. If you can, it must adapt the standards, recycle the standards, but be fair in all meanings of the word. So this ESA standard has been determined by Sansone. It's being actively developed. It's being used to actually describe an experiment. And to describe this experiment, this is just one component of this ARC. So you take ESA to describe your experiment. Now we have much more better data, metadata, which can be used. And because we have this data plant FDO concept, which we can now input out and roll out as the extended middleware for FAIRagro. And that allows us, and you see the collaboration, there's always been collaboration. So this was originally published by Data Plant, and the same figure was picked up by FAIRagro because this is the way it can be done. So you can access this. You have the ESA tab format to explain how your experiments were done. You have common workflow language. So if you actually want to evaluate things, you want to talk to Cywin, or in Data Plant, it's with Galaxy, a very useful infrastructure to talk and use data. You can actually edit your data. It's a GIST version, and you can register this. Afterwards, if you're done, you can have a data publication, and Data Plant is pushing this ahead, that this gets more and more credited. You can put it into endpoint repositories, like I told you, these domain-specific endpoint repositories, whereby you can actually take your data and put it into domain-specific SINs. And of course, you can find it with Google, which is probably most important for many, many users that are not so data centric. But of course, we have a big user base, and we have to make our big user base happy. And this is the one way to do this. And of course, there's many, many more ways to find your data then. To do this, Data Plant has this ARC RO-crate profile, which is a FAIR digital object. So it has also different ways, and actually all the software is written. So this is, I guess, a fantastic case to show that the NFDI is really working together, and that it's a good idea that we have things starting at different points in time. So it can actually be hosted as raw data, but also be converted to JSON LD, which is important to get this, you can get the specification at this ArcRDM website. You can describe this, and you must describe this ESA. If you have workflows, you put this in, and then you have all these different ESA parameters inside with which you can check your data. To just give you an example on how that works, what does it mean? This ESA, it stands for Investigation, Study, and Assay. And it was meant to actually be simple for biologists. So you have an investigation. What do you investigate? You have certain studies inside. For example, you work on different plants and you sample the plants. So this is called an assay, where the different assays describe things. And you can link this, and you should link this to standard operation protocols if you have them. But in many labs, especially in molecular field, you might have that. However, also, of course, if you go to a larger scale, of course, you work on plots in the agri-domain. So you have your big field, you have different plots, and you measure them in a standardized way. And you know the standardized way. And you describe this to your colleagues when you do that. And then you have the data inside. The data is coming as raw data, but it can also be processed. So the raw data, you might measure this with a sensor. But of course, if you have raw sensor data, you transfer the sensor data to something, and you get your measurement data out. At the same time, you have, of course, your material. And of course, this is not only for plants. It was meant to be much broader. It can be in the life sciences, but also even beyond that, you can apply this to process and engineering. You can apply this to processes in computer science, whereby you have this, and potentially we just have to rename things a little bit to make this a bit more obvious. But it actually gives you the advantage that you can completely describe your process to going from one thing to another. Now, this was developed by Dataplant. And then you might ask, so we are on a FAIRagro talk now. What's the difference? Dataplant was more on plants and basic sciences and looking at smaller objects, whereas I told you that FAIRagro is going all the way from the gene to the region. Dataplant is more down here. FAIRagro is all the way up to here. So it's actually a fantastic collaboration because Dataplant has developed something as a structure, which can now be applied as this FAIR digital object to go ahead and actually show and explore how this can go beyond its immediate development and how it can serve as a way for the to drive the middleware of FAIRagro ahead and beyond. And of course, for this, there's long ongoing discussions with many, many meetings already to actually adopt this and to go ahead. Of course, Dataplant could capture all of this already, but Dataplant was based on smaller sample sizes. So we did not have in Dataplant georeference data so much. Now looking from the FAIRagro side, we come from the top and we ask, yes, we have that, but how can we bridge this? Is this in there? How do we actually have the metadata to support all of this? And to bring this together and to make data fairer, there's a common thing now that actually this Arc Data Hub works and actual real field data and real data from FAIRagro is put into that because, of course, in the theory, everything works. In practice, of course, somebody actually needs to work on the data and actually somebody needs to do it. And by that, we will figure certain things out and we will figure out if there's cornerstones that we can tackle jointly. And that will be the first case to broaden this even further. With other life science domain, of course, this is simple. For example, if you work in microbiota, it's also a smaller use case. But FAIRagro is a fantastic case because in FAIRagro, we look all the way from region all the way down to individual plant, of course, mostly not in one experiment. But now in the future, we can. What will the individual plant contribute to the geosphere of this different plants? And with this, I thank you for your attention. I thank you for staying here with us. And despite the summer holidays for many of you, I hope I have shown you that FAIRagro is dealing with a broad case, but is now can stand on the shoulders of Dataplant and can actually develop this FAIR digital object ahead and actually use it reuse it, and send it for data analysis to make data absolutely more fair, and not only fair, but with little interoperability, by increasing this, by integrating the legacy data. And I'm very happy to take your questions.
Thank you, Björn, very much for this great introduction on the efforts of DataPlant and FAIRagro. And now, before we start with the question and discussion session, we say goodbye to our viewers on YouTube. The next NFDI talk will, right now, the NFDI talk will make a summer break, and the next talk will take place on September the 7th. And it will be about giving an introduction to the base for NFDI basic service, NFDI.software. We hope to see you there after the summer break. So I will now stop the stream to YouTube. This will again take a second.